Today we are going to learn about electron microscopy. The electron microscopy is one of the important technique in nanomaterial characterization. In today's lecture, we will be learning about the theory and principle of scanning electron microscopy and transmission electron microscopy. By practical demonstration, we will also learn this technique more in detail. Let us see what is scanning electron microscopy. Scanning electron microscopy is an imaging technique that uses a focused beam of electrons to provide high resolution images of the surface morphology of materials. SEM is highly utilized in the fields like material science, biological science, and nanotechnology for material characterization. So, these are the various parts of scanning electron microscope. The first one is the electron gun. So, that is the source of electrons. Followed by we have the condenser lens, which focus the electron beam. We have the objective lens, which further narrows the beam to focus on this sample. We have the scanning coil, which moves the beam across the sample, and we have the sample chamber where sample has to be placed under the vacuum condition. Once the signal is generated from the sample, so that will be detected by the detectors. So, we have three detectors. One is backscattered electron detector, X-ray detector, and secondary electron detector. The signals will be amplified and the image will be generated. So, let us see the working principle of scanning electron microscope. The scanning electron microscope, that is SEM, utilizes a focused beam of high energy electrons, and it has a magnification of 20 to 1 million times. The principle is electrons interact with atoms on the sample surface, which will generate various signals such as secondary electrons, backscatter electrons, and X-rays. And the detectors collect these signals generated from the sample, and it will provide the information about the sample. For example, it will provide the information like surface topology, composition, and physical properties of the your sample. How do we get image from the scanning electron microscope? Here the first step is the electron beam is generated from the electron gun, and the beam is focused on the sample by the electromagnetic lenses. The beam scans the sample, and the signals, that is electrons emitted from the sample, that will be detected by the detector, and image will be constructed on the screen. So, it will scan the complete sample, and finally you will get the image that will be generated in the your computer using the software. So, let us see what are the various type of reflected electrons and what kind of information we can get it from these reflected electrons. The first one is secondary electrons. From the secondary electrons, we can get the information like surface topography and fine surface details. And from the backscattered electrons, we get the atomic number contrast and material composition of your sample. And third one is the X-rays. By using that, we get the elemental composition and elemental mapping. So, let us see this in detail one by one. So, let us see what is EDX. EDX is energy dispersive X-ray spectroscopy. EDX is a technique used for the elemental analysis of the sample. So, what is the principle? The principle is X-rays are emitted due to ionization of the sample upon interaction with the electron beam, and these X-rays are unique to each element. So, this will help us in identifying and quantifying the each element present in the particular selected area of the sample. For example, in this picture, if this area is selected and this area, we can understand what are the different elements present in this particular selected area. In this case, we can see that carbon, which is 70% weight, and oxygen, which is around 29% by weight. By this technique, we can understand the elemental composition of the particular sample in the selected area. And next one is elemental mapping. This elemental mapping is a technique used to visualize the spatial distribution of elements within the sample. In this case, we can select the particular area in the sample, or the complete sample can also be analyzed for the spatial distribution of various elements within the sample. In this EDX data, we can understand the elements are represented in the color coded maps. For example, here the red color denotes the carbon, and this yellow color denotes the nitrogen, and the green color denotes the oxygen. So, this will give you an overall idea about what are the various elements present in the particular sample. Let us see the difference between the SEM and FESEM. The main difference is the electron source. In the scanning electron microscope, we will be using the thermionic electron gun, which is a hot emission. In the FESEM, we will be using the field emission gun. This is a cold emission. And in the FESEM, we get the higher resolution, but it requires ultra high vacuum. Whenever we are using these biological samples or non-conductive samples, we get poor resolution and sometimes the sample may get damaged. So, how to overcome that? We can overcome that by sputtering of conductive materials. How we can do that? We can do the sputtering of conductive materials like gold, which will enhance the image quality. How it is increasing the image quality? It will increase the image quality by increasing the conductivity. So, once the conductivity is increased, the secondary electron signals are emitted more efficiently, which results in sharper and higher resolution image with improved contrast. And this fine grain structure of coatings provides a smooth and uniform layer which will enhance the fine details of the sample surface. In case if you are using a non-conductive samples, these coatings create a conductive path for excess electrons to dissipate. So, that will reduce the sample surface charging and it will improve the image stability. So, let us see the difference between the uncoated and gold coated sample. So, this is a non-conductive sample. When you are using the uncoated sample, you can see that the resolution is poor and the sample get damaged. And when you are using the uncoated sample and if you are using the low voltage or low vacuum, the resolution is still poor. The same sample when you are coating with the gold, you can see that you get a better resolution and you get a better quality images. So, that is the advantage of this gold sputtering on the samples. Let us see some of the important factors for conductive sputtering. The first one is the coating material. We can use the gold, platinum, silver or chromium, depends on your sample. So, gold is the popular choice because of its chemical inertness and high conductivity. And the thickness of the coating also plays a very important role. Usually it should be between 2 to 20 nanometer and it depends on the sample as well as your desired resolution. And the grain size of the gold coating should be in the range of 8 to 12 nanometer. If you are using the lower sputtering current, which can produce a smaller grain sizes and dense layers, and high vacuum results in the finer grains. So, these are the parameters we have to follow when we are doing the conductive sputtering for getting the better image. Let us see the relation between voltage and image quality. Whenever you are using the higher voltage, you get the better resolution, but there is a chance your sample may get damaged if you are using some biological samples or polymeric sample. And in the lower voltage, it reduces the sample charging and it also reduces the damage, but again the resolution will be very low. We have to use the optimal voltage settings to improve the image quality. Let us see some of the challenges or problems in the scanning electron microscope technique and how to overcome that by some of the troubleshooting tips. The first problem is charging of sample. Charging means it is the accumulation of electric charge on the surface of non-conductive samples when sample is exposed to high energy electron beam. And these non-conductive materials cannot dissipate the electrons efficiently, so that leads to charge buildup. So, this leads to cause image artifacts or bright spots on your image. So, how to overcome this? By applying a thin conductive coating. You can use the gold or palladium, or you can use the low vacuum SEM mode, or you can reduce the beam voltage. By doing that, we can overcome the charging of sample. From this picture, we can clearly understand when you are using this excessive charging, the sample is getting damaged. So, the excessive charging on the polymeric nanoparticles, it causes damage and the burning of the sample. The next important challenge is moisture in the sample. If there is a moisture in the sample, that will evaporate under the vacuum condition of SEM, so that leads to physical changes such as shrinkage or deformation. And the interaction of water molecules with the electron beam, it can disrupt the path of electrons, which leads to unstable focus and poor image quality. And this moisture can also result in additional charge accumulation due to interaction with the electrons, especially in case of non-conductive samples. And poor vacuum condition arising due to water evaporation can scatter electrons, reducing the focus of the electron beam. How to overcome this? We can dry the sample thoroughly using IR lamp or in hot air oven, or you can store the samples in the desiccator. From this picture, we can clearly understand the moisture in the sample causes a change in the surface morphology of the sample while imaging. Let us learn how to prepare sample for the SEM analysis. The first one is you have to clean the stub. Clean the surface of the stub using isopropanol or ethanol. So, this is the stub, we have to clean it with the isopropanol or ethanol and apply the conductive tape over the stub. If it is a liquid sample, cut a glass slide into 1 centimeter pieces, stick the glass slide using a conductive tape on the stub. You can add the nanoparticle solution on the top of that. You can add few drops of the sample on the glass slide, irradiate the sample under the infrared lamp for drying it. Once the sample is dried, keep the dried sample in a desiccator to avoid the moisture. Then coat the sample with gold or other conductive materials to get the better image. And these are the gold coating parameters. We have to maintain these parameters to get the better quality images. The next one is solid sample. If you are using a solid or powder sample, the dried powder sample can be placed on the conductive tape using a spatula, and it has to be evenly spread over the conductive tape. Then remove the excess powder by blowing the air over the sample. Then coat the sample with the gold or other conductive material. Whenever we are doing this second step, avoid pressing the sample with the spatula to prevent disturbing its original morphology. And third one is, if you are using the solid, that is the whole sample, in this case you can stick the solid sample on the stub using the conductive tape, coat the sample with the gold or other conductive material. Once the gold coated sample is ready, place the prepared stub in the stub holder and insert the stub holder into the SEM machine and start imaging. You will get this kind of final image. This is a mammalian cell. You will get this kind of image in the scanning electron microscope. I hope you understood the basic and theory of the scanning electron microscope. The next technique is transmission electron microscopy. The transmission electron microscopy is a powerful technique that uses a beam of high energy electrons that is transmitted through an ultra thin sample to produce the detailed images. So, here the electrons are transmitted through the sample. So, that is why it is called as transmission electron microscope, and this TEM provides a near atomic resolution, and it will be very useful for understanding the structures at the nanoscale. It also gives the information about the size, shape, morphology as well as the crystallographic information and composition information of the particular material. So, let us see the various parts of TEM. The first one is the electron gun, so which is the source of electrons. In the TEM we will be using thermionic or field emission gun, and the next one is the condenser lens, which will focus the electron beam onto the sample. Then we have the specimen holder, which holds the thin sample in place under the vacuum condition, followed by we have the objective lens, which forms the initial image by further focusing the electron beam, and we have the projection lens, which will magnify the transmitted image before it reaches the detector. And we have the imaging detector to capture the image, and we have the spectroscopy detector for elemental analysis. Let us see the working principle of the TEM. Here the electron gun generates the high energy electrons, then the electron beam passes through the thin sample, then electrons are either transmitted through or scattered or absorbed by the sample. Then the magnetic lenses focus the transmitted electrons onto a phosphor screen or a CCD camera to generate the image. Thicker or denser areas scatter more electrons and appear darker. From this picture you can clearly understand. So, this is the core shell nanofiber, this is your core and this is the shell. The core is loaded with the anti-cancer drug, so that is why the core is appearing darker when compared to the shell. Let us understand about the diffraction patterns. That is, SAD is a selected area electron diffraction. SAD is a crystallographic technique used to analyze the crystal structure of the selected area of the sample, and these patterns provide information about the atomic arrangement and orientation of the material. These include spot patterns. If it is your sample is a single crystal, you get the spot patterns, and if your sample is a polycrystalline, you get the ring patterns, and if the sample is amorphous, you get the diffuse halos. So, let us see the diffraction pattern with some of the examples. The first one is crystalline materials. The diffraction pattern is made up of distinct spots. These spots corresponds to specific crystallographic planes in the sample. The arrangement and symmetry of the spots can reveal the crystal symmetry and orientation of the sample. For example, in case of single crystal gold, you get this kind of distinct spots. So, let us see the next example, that is the polycrystalline material. When the material is made up of many small crystallites, it gives rise to concentric ring pattern. The sharper and more distinct the rings represent the larger grains. The diffuse rings indicates smaller grains or amorphous material. The spacing between the rings provides the information about the distance between the atomic planes, which can be used to determine the lattice parameters such as edge length and edge angles. If you are having a single crystal, you have this kind of dotted pattern. If you have a polycrystalline material, we will have this kind of dotted pattern. So, this is an example, the polycrystalline aluminum. If you zoom this image, you get this kind of dotted pattern. The next example is amorphous material. For amorphous or non-crystalline materials, a broad diffused halo in the diffraction pattern will be obtained. The halo depicts the lack of long range order in the material structure. From this picture, you can understand this is the amorphous quartz glass. You can see the diffused ring pattern. So, let us see some of the common problems encountered while doing the TEM and how to overcome these problems. The first one is sample drift. During imaging, the sample may shift due to thermal or mechanical instability, due to which you get the blurred image. The second reason may be contamination. Hydrocarbon deposition from the vacuum system can contaminate the sample. The third one is radiation damage. The high energy electron beam can damage the biological or some of the sensitive sample. The fourth one is the charging effects. If you are using a non-conductive samples, accumulate more amount of charges, which will leads to artifacts and poor image quality. The fifth one is the vacuum leaks. If the TEM operates under high vacuum, there is a chance the vacuum can leak and lead to poor image quality. So, how to overcome these problems? If there is a contamination, clean the sample holder and vacuum system regularly. Use the plasma cleaning to remove the hydrocarbons. If there is a damage due to radiation, lower the beam intensity, or if you are using a very sensitive sample, temperature sensitive sample, you can go for cryo TEM for the sensitive samples. And to overcome the charging effects, we have to coat the sample with a thin conductive layer. To overcome the vacuum leaks, we have to regularly inspect the O-rings and vacuum seals in the grid holder. And to overcome the sample drift, we have to allow time for thermal stabilization, or we can use the software and correct the drift in the software. If it is a low contrast sample, if you are using some biological samples or some of the polymeric nanomaterial sample, to overcome the low contrast, we have to stain the sample with heavy metal salt. For example, we have to use the uranyl acetate or phosphotungstic acid to enhance the contrast. Here you can see this is a negative staining of dendrimer nanocarrier. So, negative staining means you will stain the background and you can see the nanoparticle. So, let us see the difference between the TEM and HRTM. So, the main difference is the resolution. In HRTM, that is a higher resolution transmission electron microscope, you will get sub-angstrom resolution and you get ultra high magnification up to several million times. But you need ultra high vacuum for this atomic resolution, and this HRTM plays a very important role in the nanomaterial characterization, especially in case of if you are using these small size carbon dots or nano clusters, the HRTM will be very useful. Let us see how to prepare the sample for the TEM analysis. Once prepare the nanoparticle, dilute the nanoparticle in the appropriate solvent, then drop cast onto a TEM grid. The TEM grid is placed on a filter paper and you can add a drop of your nanoparticle solution. Then you have to dry it overnight in a desiccator. The excess nanoparticles solution can be absorbed by the filter paper. This is called drop casting method. And this is a TEM sample grid. Here the TEM grid will have layers. The first layer is the carbon, the next one is formware, and the bottom is your grid. The thin carbon layer on the top of the formware layer of copper grid, it will dissipate the excess electrons and stabilize the sample. And this carbon has a low electron scatter, so it does not interfere with the material image, and which will help in the getting the better image. Let us see how to prepare biological sample for the TEM analysis. The first step is fixation. The biological sample should be fixed using glutaraldehyde or paraformaldehyde. Then followed by that we have to dehydrate it using ethanol or acetone solution. Then you have to embed the sample using the suitable resin. Followed by that we have to do the sectioning, that means you have to make ultra thin sections using the ultra microtome. Followed by that you have to do the staining. So, as I told earlier, biological samples or some of the polymeric samples, we have to stain with the heavy metal stains such as uranyl acetate or lead citrate to improve the contrast. So, this is called negative staining. That means it stains the background so that you can get the better quality images. I hope you understood the SEM and TEM. Before we go to the practical demonstration, let us briefly understand the difference between SEM and TEM. The operating voltage in SEM is 0.5 to 30 kV and in TEM which is 80 to 300 kV. Here in SEM we get the resolution of 1 to 10 nanometer. In TEM we can get 0.5 nanometer to sub-angstrom resolution. The application is in SEM, it can be useful for surface morphology and to detect the material composition. In case of TEM, we can get the information structural and crystallographic analysis, and also we can go for even atomic scale imaging. I hope you understood the theory and principle of the SEM and TEM. Let us go to the lab and learn this technique more in detail. We are going to learn how to perform a scanning electron microscopic analysis. BSA nanoparticles, SEM stubs, glass slides for drop casting the material, glass cutter to cut the slides, pipette, tips, carbon tape, forceps, beaker for discarding the waste, tissue paper and ethanol for cleaning the glass slides. First, we are going to serially dilute the stock solution of the BSA nanoparticles to get a proper image. We will prepare 1 by 10th, 1 by 100 and 1 by 1000 dilution of the stock solution. So, we will add 1 ml of the stock solution to 9 ml of ultra pure water so that the final dilution will be 1 by 10. Mix it properly. Similarly, using a fresh tip, we will add 1 ml of the 1 by 10th dilution to a 9 ml of ultra pure water so that the final dilution of this solution would be 1 by 100th. Similarly, we will take 1 ml of 1 by 100 dilution and add it to 9 ml of ultra pure water to get a final dilution of 1 by 1000. We are going to follow drop casting method. For that, we need a glass slide and we have to cut it into 1 cm by 1 cm. We can use a glass cutter to cut the slides. Carefully cut the slides into 1 cm by 1 cm. Now, we will drop cast the sample onto the cut glass slide using micropipette. We need a IR lamp to dry the sample. Carefully add sample, ensuring that it does not flow out of the slide. Now we will dry the sample for about 10 to 20 minutes. We can see that the sample has completely dried. Now next step is to place the glass slide on the stub. Stick a piece of carbon tape on the stub. It is a double-sided conductive tape. Remove the white cover using a forceps. Hold the glass slide carefully at a corner and stick it on the stub. Tap it in a corner to secure it properly on the stub. This is a sputter coater which we will use to coat our sample with a thin layer of gold to make it conductive. Select proper sputtering settings. Now place the stub in the machine and start the process. We are using 20 milliampere of current and 45 seconds of sputtering time. You can see that the sample has been coated with a thin layer of gold. This is a scanning electron microscope which we are going to use to analyze the surface of the sample. First step is to vent the chamber, then place the stub in the sample holder and close the chamber. After that, we will pump the chamber so that vacuum is formed inside the chamber. After the vacuum has formed, we will move the sample holder. Adjust the height of the stage accordingly. Use the screen on the bottom left to navigate around the sample. We need to bring it to appropriate working distance. After that, we will first adjust the voltage and magnification and then turn on the beam. Select the appropriate detector. You can see that the image is starting to appear on the screen. We will increase the magnification and then we focus on a part of the sample. The focus and contrast are adjusted till we get a clear image of the sample surface. We can see that the sample surface is getting scanned and sharp image is forming on the screen. You can save this image in the folder with proper sample name and date. This image depicts the surface morphology of the synthesized BSA nanoparticles done using a scanning electron microscope. In 1/10 dilution of the stock solution, you can see the densely packed nanoparticles. Upon further dilution and magnification, individual nanoparticles can be observed with sizes falling within the nanoscale range. For TEM analysis, we have 1/100th dilution of BSA nanoparticles and we need TEM grids. This is the box in which TEM grids are kept. We require a special forceps with very fine tip to hold the grid. You should be very careful while handling this, and we will need filter paper for sample preparation. So first we will take out a new TEM grid. You can see that it has two sides, one shining side and the other one is carbon coated non-shining side. We have to place it with the carbon coated non-shining side facing up. Then put a drop of the sample solution on the TEM grid. The excess sample solution will be absorbed by the filter paper. Use an IR lamp to dry the sample, or we can also dry the sample overnight in a desiccator. Once it is dried, we will put the TEM grid in the TEM sample holder. This is a sample holder for TEM. First open the latch . And then carefully place the TEM grid on the sample holder. Close the latch to secure the TEM grid and tap it a few times to ensure that it is properly fit. You can see the TEM sample holder with the grid at the tip. This is the transmission electron microscope which we are going to use it today for analysis of our sample. The sample holder will go inside through this port. We will insert the sample holder in the sample port. And then pump the chamber to form ultra high vacuum. After ultra high vacuum is achieved, the sample holder is inserted further into the machine and rotated into the closed position. This is a setup to control the machine. The voltage is set and the beam is then turned on. The magnification and focus are adjusted to get a clear image of the sample. You can see the TEM image of the BSA nanoparticles on the screen. Finer adjustments need to be made to get a clear image. Now we move on to the SAED pattern. The beam stop is inserted over the sample. And the beam is focused on the center area. Then the diffraction pattern is recorded. The diffused rings denote the amorphous nature of our sample. As a summary, in today's lecture we learnt about the principle and theory of SEM and TEM, and also by practical demonstration we understood these techniques in detail. Thank you for your kind attention. I will see you in another interesting lecture.